The other part of the cell is cell nucleus, that in this session we want to know about the microscopic structure of the cell nucleus, type of division, and cell cycle. You see here the structure of the nucleus that have a membrane that named nuclear envelope. There are nuclear pores inside of it, and in the internal part of the nucleus there is nucleolus, and it includes the DNA. Nucleus contain the DNA or chromosome, and it's also, or additional to DNA, it includes some molecular machinery that includes some transcription factor or other factor that to replicate DNA and synthesis the process of all type of RNA. The nucleus in the mammalian cell consists of the nuclear envelope, chromatin, and nucleolus. Nuclear envelope or nuclear membrane consists of two concentric membranes that's separated by nuclear or perinuclear, perinuclear space, and in the nuclear envelope there are nuclear pores. The outer membrane of the nucleus is continuous with rough endoplasmic reticulum. It can and also associate with ribosome, that these are reason for economy of the cell, that because the ribosomes synthesis in the nucleus, they should transfer to RER and also to cytoplasm for production of protein. The inner membrane of the nucleus associate with nuclear lamins. If you memorize lamins of one type of intermediate filament that are in all of the cell nucleus and help to maintain of the shape of the cell and also for formation of the nuclear pore. Also, the inner membrane is associated with the chromatin and also ribonucleoproteins. Nuclear pore, this structure composed of a central cylindrical body that placed between the inner and outer membrane of the nucleus and consists of 8 nucleoporine proteins and are lipid-free. Yes, in the pore, yes, include 8 of the nucleoporine, that is one type of transmembrane protein. Also, in the central cylindrical, cylindrical structures also have 8 spokes, that these spokes in the inside of the nucleus are related to the lamins. And finally, these pores are necessary for trafficking of the nucleocytoplasmic process. Chromatin is a hereditary material and include the chromosomes, that the chromosomes include the DNA and some proteins. Chromatin divided based if its activation to heterochromatin and euchromatin. Heterochromatin is more coiled and it's not activate in the transcription and mostly located in the venous of the cell membrane. Against of the euchromatin, if you memorize, EU means good. Euchromatin is a good chromatin that involved in the transcription and mostly located in the center part of the nucleus. Schematically, by electron microscope. The dark part is a heterochromatin. They are dense and transcription not occurred in them, but the euchromatin mostly located in the center part and they are involved in the transcription. The cells that are very active, such as neuron, most of their chromatin is euchromatin. Maybe you hear that the neuron are euchromatin nucleus because all of their chromatin is euchromatin. Heterochromatin again in the cell divided in two types. One type is a constitutive and another type is a facultative. Constitutive heterochromatin condense throughout the cell cycle and development, and their function mostly is structurally. But the facultative heterochromatin can convert to euchromatin and also euchromatin converted to heterochromatin state, and they has the potential for gene expression in the development or in cell cycle. But the constitutive, such as telomeres, have a structural function, and but they also replicate in the space and reduce in the recombination. Nucleosome is a subunit of the chromatin. Each nucleosome includes 4 pairs of the histone that are H2A, H2P, H3, and H4, that they form the nucleosome, that they coiled one DNA chain around themselves, and by this function they condense DNA and coil the DNA. And the DNA by another histone that named H1 coiling and rounded helically around the other histone. It means that these 4 pair of histones form the core of the nucleosome, and H1 histone link them together. Yes, here you can see the DNA. If we see ultrastructurally, we can see that in the DNA include some nucleosome, that these nucleosome include the DNA chain and also histone, core histones, that are these 4 histones. The high amount of the nucleosome packed together and form the chromatin fibers. Finally, they extended some loop transcriptionally and form the chromatin. Chromatin have 2 types, heterochromatin, euchromatin. Heterochromatin condense, but euchromatin is disparate. And finally, the chromatin form the chromatid, and finally chromosome formed by a pair of the chromatid. As you see here write the diameter and size of each of them, that don't need to memorize you them. Thus, briefly, the DNA smallest subunit is nucleosome, that each nucleosome includes the one DNA, that each DNA includes the 152 base pair, and also core of the histone molecule include H2A, H2B, H3, and H4. That H1 link them together, and this part of DNA is a linker DNA that link the DNA to H1. Nucleolus is a spherical structure and highly basophilic structure that present in the nuclei of the cells and active in protein synthesis. It's a basophil structure because of the presence of the concentrated RNA that transcript, proceed, and complex into the ribosomal subunit in nuclear region. The molecule of RNA also synthesized and modified in the nucleolus very quickly, and the newly organized small and large subunit of the chromosome, and also rRNA exported back to the cytoplasm. If you memorize in last part, I explained for you that the proteins that are necessary for the organization of the ribosomal subunit and synthesis of the RNA synthesis in the cytoplasm. They come to the nucleolus, involved in the synthesis of the RNA and ribosome subunit, and finally these products transfer again to cytoplasm. Here you can see the electron microscope of the nucleolus, heterochromatin in the thinness of the nuclear envelope, and euchromatin. And here you can see one neuron that, because the most of their chromatin is euchromatin, the nucleus is very colored. And this nucleus named euchromatin, and this structure is a